Examples of autocrine, Juxtacrine, and Paracrine signaling by immune cells. Autocrine. Here we see an antigen-presenting cell and a naive T cell. The naive T cell comes in contact with the antigen-presenting cell, and its receptors bind to the antigen. The T cell is now activated. There are many receptors on the T cell for interleukins. Interleukins are a type of cytokine that serve as a chemical messenger. They can cause cell proliferation, differentiation, and activation. After being activated, the T cell releases its interleukins. The T cell itself has receptors for the interleukins that it is releasing. The interleukins bind to the T cell's own receptors. What happens when the interleukins bind to the receptors of the T cell? The interleukin serves as a ligand, and when it binds to a receptor, it causes a signal transduction pathway that eventually results in gene activation. This gene activation can result in proliferation, making the T cell grow and divide to make more T cells for a faster immune response. Autocrine signaling is when a cell releases chemical signals that affects itself. Notice how the T cell releases interleukins that bind to the T cell's own receptors and cause proliferation. That's autocrine signaling. Juxtacrine signaling. Here is a cytotoxic T cell. Its primary function is to identify and destroy cells infected with a pathogen. When the cytotoxic T cells come in contact with an infected cell, it recognizes foreign antigens on the surface of the infected cell. The T cell immediately begins to release toxic granules containing many enzymes targeted at killing the infected cell. The granules travel a very short distance since the two cells are so close together. There are many enzymes in the granules. Some of the most important enzymes are perforin, which forms pores in the infected cell's membrane to allow other enzymes to enter, and granzyme and granulosin, which cause a cell reaction that eventually leads to apoptosis. Apoptosis is programmed cell death. The infected cell eventually dies of apoptosis and cannot serve as a host for the pathogen anymore. The cytotoxic T cells look for other infected cells to eliminate. Juxtacrine signaling is when cells interact through direct contact, exchanging short-distance signals. Notice how the cytotoxic T cells release its toxic granules once it touches the infected cell. The granules travel very short distances to reach their target. Paracrine signaling. Here is a naive helper T cell and an antigen-presenting cell. The helper T cell comes in contact with the antigen-presenting cell and recognizes foreign antigens on the antigen-presenting cell with the help of CD4 and MHC class I molecules. After recognizing the antigen, the helper T cell begins to produce many cytokines. These can be different types of interleukins, etc. Cytokines control the growth and activity of many types of immune cells, such as B cells, macrophages, T cells, etc. Let's look at what happens when a cytokine binds to a B cell receptor. Note that cytokines affect many types of cells, not just B cells. When the cytokine binds, the B cell undergoes many cellular changes. The B cell divides into plasma cells, which secrete antibodies, and memory B cells, which help the body fight future infections. Paracrine signaling is when one cell releases many signals that affect nearby cells. Notice how the helper T cells, once activated, releases many cytokines that affects many different types of cells, such as B cells, macrophages, and other T cells. Thanks for watching!